core of presentation where all people involved directly in the activities will present specific topics where they are involved. So it's a pleasure, a lot of things to discuss, not so much time, so let's start. Before starting, I would like to ask to the first three speakers to join me on the stage, so please. So the first three speakers are Professor Giuliano Langella, please. Thank you. Good morning again. The Associate Professor Roberta Paradiso. Thank you. And Associate Professor Alessandro Vannozzi. Thank you so much to be here in presence. So today we have a blended format. Someone is here in presence. Others joined us in streaming. So I hope everything will go smoothly. So thanks all to the staff that did a great job up to now, and I hope this will be also in the next 3 hours. So thank you. Let's start. Agritech is one of the 5 national research centers full focused on advanced technology in agriculture. So we have 47 partners that in 2022 funded our foundation, this center. So less than 3 years ago we started. This partnership covers all Italy, as you can see, from north up to the south. We have a combination, a blended composition of our organization that is structured in hub-spoke format, where hub is the manager and drive the entire project, and spokes leads research and coordinates all activities in terms of technical aspects and administrative aspects of the project. So this blends public and private is a key point because of this allows to say, let's say, that the business needs drives research. So this is an important aspect that is typical of our positioning, the center, I mean, that is the link between the research and the industries. So our partners are private partners, covers all aspects of the main aspects of the research. Some numbers here. So the project it counts in an amount of 350 million euros and 350 million euros, and 320 are full funded. We have 9 spokes that drives research that are distributed along Italy. 3 are in the north, 3 in the center, and 3 in the south, I mean. A lot of activities are carried out into the spokes. Here you can say that the spokes are split in work pack and task. Consider that the research activities inside the spoke now are around 300. A lot of people work in working groups. We have a little bit less of 100 working groups that use more than 2,000 researchers in the activities. So it's a huge project, needs a structured organization, and considering the timing for the project to complete all the activities, this is a real strength, a good challenge. So what we do, so if we talk about technologies, the activities are quite spread. I mean, so we come from sensors up to software. So we have solutions that are hardware, others software. Sometimes we go into a specific study that for strategical topics in terms of biology or genetics. So all the disciplines into the business, agricultural business are covered. The single technologies are the building blocks for our business context. I mean, the added value of this center is that we can integrate these technologies to produce advanced technologies that can solve complex problems. So our focus is to integrate all these technologies to give, to deploy to the business context solutions of real problems. Now, what the hub is doing, I mean, hub is full focused on management, training, and technology transfer. Let's say that management is the key because of this complex organization. Then training is part of our main task, let's say joined with dissemination, because of this allow to work on increasing culture on advanced technology. Then technology transfer is the final target, because we have to cover the gap between the research and industries. And final target is to build the bridge from university to the industry. Now, main challenges. Here, we have, in front of us, we have a general transition, digital transformation, technological gap, new technology introduction, sustainable strategies. So all of them could be, from one side, can be seen as real challenges. In my opinion, in front of us, we have a great opportunity, because the general transition, the generational transition and the digital transformation allow us to approach the introduction of new technology with different people, with different educational culture that is more receptive, I mean, than before in terms of introduction of new advanced technology. In which way the hub is going to approach these challenges? I mean, we have 5 pillars. For sure, we are working on founding ops, because this is the feed of new research. Then we have training, dissemination, technical consulting, technology brokering, that is part of the how can I say, working on culture that allow the new technology to be implemented, because most of the companies that we are approaching are not ready to introduce new technologies. So we need to cover 2 gaps. I mean, the first is to cover the first step of technologies, even if this means in working on introduction of traditional technology. Only when we complete this step, we could be ready to the next step, to go ahead with the next step, introducing the advanced technology. Advanced technology is something that makes our companies ready for competition, global competition. So, in doing that, we are looking for cooperations. 2 are the main areas where we are working to. The first is technology transfer, as we said, and the second is integrated solution. In which way we intend to approach these 2 cooperation with an international outlook. For sure, technology transfer, means find someone that can work with us in mutual development, participation to project, and custom training. These are, this is a not limited list, but this is what we are going to look for in our preliminary, in the next short terms. I mean, what about integrated solution? We can work together in advanced and applied research, because we can develop in the new projects with together developing research, specific application, specific technology with new partners, enlarging, I mean, our partnership. And then we can have, if these partners are international, we can work with mutual field tests, because different real condition, and this allows to test the current available technology in different environment, and of course multidisciplinary solution co-design, because we can look forward and develop some advanced technology not already into the project. So I hope that this short introduction give to those of us that are not familiar with the project to have a general idea about what we are doing with this project. Then I would like to enter now into the specific task. Please confirm if we have online Raffaella Balestrini, that is the spoke leader of the spoke number 1. So if she's online, I introduce a little bit. Raffaella. Anyway, so Raffaella. Can you hear me? I am online. Perfect. Thank you. Thank you, Raffaella, to join us. Good morning, and thank you to waking up so early this morning. We have 7 hours behind in Italy, so thank you again. Happy Easter. So Spoke 1 is mainly focused on plant, animal, and microbial genetic resources and adaptation to climate changes. Raffaella is a research director at CNR, that is one of our national research center, and director of the Institute of Bioscience and Bioresources of the CNR. So thank you again, Raffaella. The stage is yours. Stay in 15 minutes if you can, and thank you again.
Yes. Okay, thank you very much, Marco, for the introduction. And Spoke 1 is the Spoke more related to fundamental research. And Spoke 1 is aimed to study agricultural and forestry genetic resources at different levels to identify traits useful for adapting to climate change and improving the environmental sustainability of agricultural systems. And in these Spoke activities have been focused on a panel of crops, including forest trees, horticultural species, fruit crops, cereals, microorganisms, and also animals. And before to start with some of the results that we have obtained in these years, I would like to say that we use and we are using innovative tools to highlight, study, and exploit the biodiversity of genetic resources. Why is it so important to study biodiversity? Since biodiversity is a key component in determining the sustainability and the resilience of production systems. And this Spoke, very briefly, is divided in 3 work packages. The first work package is aimed to the identification of genetic variants that contribute to resilience and adaptation to climate change. And this is done analyzing the genetic diversity at the genome and pangenome level. And the keyword for this work package can be characterized. And then we have a work package 2, where we focus on the identification of genes, alleles, metabolites, and also morphophysiological traits relevant for adaptation. And in this work package also we look at the interaction with microorganisms, for example, at the interaction between roots and microorganisms to improve tolerance. And in this work package, the keyword can be understand. And from these work packages, we can obtain results that represent the basis also for the work in the work package 3. The keyword in this case can be breed, and in work package 3 we use and develop cutting-edge technologies and strategies to generate and validate more resilient genotypes. The main results that we can obtain from these activities are an improved in knowledge, but also improved genotypes, for example, and also optimized protocols to study biodiversity and stress response, including protocols to correlate phenotype, so what we can see, with the genotype. And starting from the results, important activities are related to the pangenome study in this SPOC. And particularly, I would like to highlight this part of research on the pangenome of tetraploid wheat. Why is it important to study pangenome? Since the high degree of genomic variation observed led to the realization that single sequence genomes do not represent the diversity within a species. And for this reason, we have to study the pangenome that represents the genomic diversity inside a species. And to this aim, we started from a large collection of tetraploid wheats, and the researchers that done this project start from about 800 accessions that were selected on the basis of their genetic diversity to construct the pangenome of this relevant crop, the durum wheat. And the accessions were sequenced using the best available technologies. And at the moment, about, about 40 accessions have been sequenced and represent the pangenome foundation. At the moment, other accessions will be sequenced to identify structural variants in the genome or small insertion and deletions with the idea to obtain data that can enrich the pangenome dataset that we have at the moment. And these data are allowing us to fully explore the huge tetraploid genome diversity for the identification of those genomic traits that are important to enhance resilience to abiotic stress. And it is relevant also to say that this part of work is done in the frame of an international consortium, including different institutions, but also companies. The same approach has been used also for other crops, for example, in other species. For example, in this case, from teff. Teff is a poorly characterized species with a high potential for food security, and it is resilient and well adapted to low input agriculture, and there is an enormous diversity of teff, but this diversity is still untapped by breeding. And in this case, we decided to use 20 teff landraces selected to represent the species allele pool and provide traits of interest. The pangenome, in fact, allow to obtain data on the core of genes that are present in all the sequenced species adjacencies, but also those genes that are present only in some of these adjacencies. And then always in work package 1, there are some activities also dedicated to obtain an improvement of existing genomic information. For example, in this work on grape one, apple, and blueberry. So also fruit crops. And particularly interesting is the fact that these researchers thought to structuring all the newly produced data into a crop knowledge base. And this is particularly important, in my opinion, also thinking at the transfer of results to stakeholders. Also in this case, I would like to note the fact that there is the word pangenome. Also in this, we can mirror the fact that this approach is really very important at the moment. And in work package 2, the idea was to understand the mechanism involved in the tolerance to environmental stresses. So the stresses due, for example, to climate changes. And also the idea is to understand why, for example, a species can grow in stress conditions and other species cannot do it, species or genotypes inside the same species also. And in these experiments, the colleagues decided to focus on tomato and to see at the recurrent stress responses. What means? Means that we have grow the plants in optimal condition, then in stress, in water deficit, then we again in optimal condition, and again in water deficit. And in this way, we can mirror better what happens in nature. We know that we can have a drought period followed, for example, by strong rains. And they focused on 2 different genotypes, since, as I have told you before, there is a variation also between among genotypes in the same species, and they applied different omics techniques, so techniques that allow to obtain a huge quantity of data, and so epigenomics, transcriptomics, and proteomics. And then, thanks to bioinformatic pipelines, they analyzed all this data and correlate all this data together. And the interesting results for the moment is it was the identification of a gene here that could be used in breeding programs through innovative techniques on which we will focus in work package 3. But in this work package, we have also dedicated time to the study of the interaction between the plant genotype and microorganism, and in this case we have focused on wheat and arbuscular mycorrhizal fungi. It is in fact already known that the interaction between roots and soil microorganism, beneficial microorganism, can lead to an improved tolerance. But what is known now is the fact that we can have variability in the positive effect due to the genotype of the plant and also the microbes, but also depending on the growth conditions, the environmental conditions. So in these experiments, we have started to use 15 durum wheat genotypes, and we have selected some genotypes in a first experiments without stress, and we have growth these genotypes in combination with an arbuscular mycorrhizal fungus, Rhizophagos intraradices, and we have observed the differences in shoot biomass, root extension, grain production, and on the basis of these differences, we have selected 8 genotypes to be used in stress experiments using water deficit, but also an excess of water, again to mirror what happened in nature. And on the basis of ecophysiological measurements, we have selected 4 genotypes that were influenced not only by the stress condition, but also by the presence of arbuscular mycorrhizal fungus, showing genotype-dependent responses and an improved tolerance. Now we are looking at the transcriptomics and metabolomics, so again, omics techniques, to verify the mechanism at the basis of this improved tolerance and to verify the determinants of this improved tolerance. But in the frame of inside the same work package also, we looked at imaging technologies, and this is particularly relevant since generally these techniques are easily applied to model plants, but not to crops. And in this case, for example, at the University of Milan, they have genetically encoded calcium indicators in tomato, and in the same time also they have developed a tailor-made microimaging system, as you can see here, to perform experiments in adult plants. For the moment, the results have been obtained in physiological conditions, but the idea now is to apply this imaging system to study the response to environmental stresses. But SPOC 1 is also dedicated to study animals and the resilience in livestock. And so starting from the study dedicated to genomics and epigenetics to support sustainable livestock production that are carrying out mainly in work package 1 and 2. We have also some activities mainly in work package 3 dedicated to artificial intelligence and computer vision systems for the evaluation of animal-based welfare indicators, or also the use of other technologies such as, for example, infrared spectroscopy to develop predictive models to estimate the resilience related traits in livestock. And also, for example, another activity is the identification of temperature humidity indexes as indicators of heat stress in dairy cows. But in work package 3, several activities are dedicated to plants and to apply innovative breeding approaches in crops to obtain more tolerant plants, climate flexible genotypes. And I would like to present this example since this group at CNR created a platform of innovative breeding approaches in grape 1. And for example, starting from soma clone regeneration, starting from Calli, embryogenic Calli, but also application of genome editing technology. And in this case, genome editing is used to obtain wine grape genotypes with improved resilience to stresses. And the idea is to select target genes whose editing can induce broad-spectrum tolerance to pathogens, rather than a tolerance or resistance to a specific pathogen only. And they have already published a method for the regeneration of edited grapes from protoplasts. You have to think that work with woody plants in this topic is not an easy task. And they have already prepared Nebbiolo lines edited for a small sequence of DNA, of RNA, micro RNA 482 that is involved in the pathogen response, and the plants will be soon transferred from vitro to soil for the characterization in controlled environment. And however, in this is only an example, since we have also other example of successful editing of plants in this spoke. And here, for example, an example using a mitochondrial gene in potato. And new plant genotypes have been developed inside this work package, but now we have to test them on large scale, and this can be done only in collaboration with seed companies and breeders. And however, it is also important that the identification and validation of the more tolerant and resilient genotypes requires an advance in technology dedicated to the evaluation of the plant status in diverse conditions. And this is also done inside this work package 3, since for example, in this case, researchers have at the campus Biomedico have prepared a multispectral device, and they have generate a pipeline through this multispectral device to verify the status of the plant. So, for example, the stress status of the plants in stress conditions. And they have compared this multispectral device with the classical devices, also, and they have already tested it also in stress conditions, particularly in salinity.
Raffaella, you have 1 minute. You have 1 minute.
Yes,
please.
yes.
Thank you.
This is last. And now the idea is to test all these new genetic materials and to develop platforms to test these. Here an example for Solanaceae mutants with improved agronomic traits, but this is valid also for other crops. And so at the end, I would like to thank you all for the attention, and all the partners involved in this spoke.
Perfect, thank you, Raffaella. At the end of each session, we are open for questions from audience, if any. If you do not have any questions, we go ahead with the second spoke. So we can proceed. So I would like to invite here on the podium full professor Giuliano Langella from University of Naples. Giuliano is professor of pedoinformatics at the University of Naples Federico II. Spoke 2 is full focused on spatial, for crop health a multidisciplinary system approach to reduce the use of agrochemicals. And today, Giuliano is going to talk about specially approach, this decision support for crop health, a scalable approach to sustainable pest management. So thank you, Giuliano, please.
So, good morning to everybody. I have to run the presentation from here. So, okay. Okay, so this is not working. Good morning to everybody. First of all, let me express my gratitude to organizers and all the staff members which allowed us to be here and share with all of you these ongoing activities we are making in these projects. In particular, my presentation will falls under the Agritech project spoke number 2. And to this regard, I'm going to show this, the progress about a flagship solution we decided to share with you in this presentation at the Expo meeting. In particular, I'm going to show you this platform embedding geospatial decision support systems dedicated to crop health. Sorry. The main goal of the activity, in particular this building of the platform, is to optimize or reduce the use of pesticides, so and the chemical fertilizers, and also promoting the sustainable practices at scale. This is made thanks to the creation of a specific infrastructure which is called geospatial cyber infrastructure, which means that we have cyber, so this infrastructure is available via the web, and geospatial means that we embed in this platform a strong geospatial component which is basically given by the geospatial data, which extends these geospatial characteristics also to models and finally to this DSS component available in the system. Some of the most important ingredients we have in this platform are, of course, the availability of decision support systems dedicated to pest assessments and forecasting, then this opportunity to have advanced and customizable user interfaces. And also it is very much important, this interoperability feature which enables this platform to communicate with other platforms and therefore exchange data and models. This is a very important characteristics that we have to point out because it's available in this platform. As you see here in this slide, what I'm focusing on is the kind of generic distinction of this geospatial cyber infrastructure in tiers. You see we have the data tier, the logic tier, and the presentation tier. Now, basically, the data tier collects and manages data about agriculture and climate data, but also user data, and of course, data models metadata, which are very much interesting and important to let these models run behind the scene. Then we have the logic tier, which basically embeds all these models according to pathogens, weeds, pests, of course enabling the prediction of risk factors for crop health. In the end, we have this presentation tier, which is very much important because it's the only way an end user can experience the usage of this platform. And over there, we have a presentation tier which offers this multi-level user experience. I'm going to show you in the upcoming slides the different user interfaces which are available. And these are available for both private and public stakeholders, which are therefore informed about the decisions they can take according to, above all, the use of agrochemicals. Now, in this slide, I'm going to show you quite the same tiers which were available in the previous slide, but using a more graphical and high-level representation of the main components involved. You see here there is the agrometeorological monitoring. We have different models according to pests, to weeds, crop. In particular, in the presentation tier, you see here this interesting distinction between the user interfaces, so interfaces dedicated to human usage of the platform, but also there is this web API, which is an application programming interface enabling the interesting interoperability feature I told you before, which is important because this component allows the platform to communicate with other platforms and exchanging both data and models, or also decision support systems with other platforms already available out there. Then we distinguish between dashboards and toolbox, and I'm going to show you in particular a demonstration in the second part of my presentation dedicated more or less to the toolbox experience, user experience. So I prefer in this second part of my presentation to share with you something more concrete. So I will jump directly to this multi-level user experience platform by means of providing you this DSS experience and workflow we have in the platform. Of course, currently the platform is offline. Why? Because we are still completing some internal functional features which are in charge of connecting different components and layers within the infrastructure. And what I did was to run this demo available on my computer and then provide you some screenshots. So what I will do basically is to share this part of the platform. It's of course something preliminary but concrete. After a registration using a digital identity, the user can seamlessly navigate the different user interfaces without the requirement of logging in again and again. As you see here, we have the first of the three user interfaces we have in the platform. In this interface, the user can manage, as you can see from the screenshot, personal data, farm data, parcel data, and also there is a section dedicated to the uploading of data related to parcels or farm. In this demonstration, I show you how I created geometry, which is identifies somehow a parcel available in Naples province, nearby Sorrento, which focuses on olive orchard available in that zone. So what I did was to upload this geometry in the system thanks to this data upload panel. And after that, what we have to consider, of course, is to populate this parcel section. So what we can do is to assign this parcel to a farm. Of course, we can have a single user can have more farm, and of course, for each farm can have more parcels. There are different information that can be provided according, for instance, to soil, but also later I will show you something also related to climate, and of course the kind of crop that is available on that parcel. Once a parcel is created, what can be done is to instantiate an automatic and continuous monitoring. Of course, this starts from selecting the parcel. So this is something working at the parcel level. As you can see here, few parameters are provided, such as starting date, ending date, and of course the kind of crop, in this case olive, we have within this parcel. Basically, to let this be operative in the system, we have to do two things. First, we have to associate a weather station to this parcel, which the system can also manage in automatic fashion. And second, we have to select which pests, pathogens, weeds we want to associate to this crop. In this demonstration, we only have one model available. As you see, we have this olive fly model, which is basically physically based models simulating Bactrocheraole population dynamics. I will show you later, the last slides, some charts according to this population dynamics. So in the final version of this platform, you will have, for a single crop, of course, more options available that we can select. So it's not only one model or one pest we can select, we have more options we can select at the same time to monitor for that single region of interest included in the system. Then, as you see here, there is a panel to manage the different monitoring operations. It's not yet available, the opportunity to click on the Show Me button, which will, in the next version, which will be available. So there will be a dashboard dedicated to the overall monitoring of the parcel, providing information about all the kind of pests, pathogens and weeds, risks which were selected by the user for his case study. What is instead available currently is the other user interface, which is called Toolbox. Toolbox is a different kind of experience that can be made within the platform, since it somehow enables the end user to build the so-called what-if modeling or scenario analysis. Why this? Because, of course, now you only find some examples of models here. You know that we only have olive fly model. But in the final version, we will have all the kind of models and DSS available within the system. So the end user can select whatever tool or model to be run, considering specific features for the simulation, such as, for instance, the user can ask what happens if I change, I don't know, the kind of the crop or the variety, the biotype, or I don't know, the date of seeding or planting and so forth. Of course, each option will create a different simulation, runtime, and of course a different output. And finally, the end user can also compare the different outputs according to the risk levels which arises considering the different options. So in that case, I will select the olive fly pest. Here you see an example of the model metadata. Then here you see that we can instantiate, according to the model execution form, we can select the region of interest, of course, the weather station or data that we want to associate with this simulation, which can be, of course, changed, and other parameters according to the kind of model we are using. When we click on Submit, what happens here is that in the results panel, we will get the list of scenario analysis or different simulations we requested. And then we can click on the Show Me button in order to get results according to different ways to communicate the outputs coming from the platform, such as tables or charts. In this case, you see an example of number of eggs, but of course also other charts and kind of graphical representations will be available in the system. So I think I can stop here. What we are going to do according to this toolbox user experience is that, of course, we have to decode this kind of information, which is related to population dynamics, for instance, to somehow a level of risk which can be easily understood by an end user. And this is an ongoing activity we are performing during these weeks. So I think I can stop here. So just to provide more time for possible discussion. Thank you for your listening and attention.
So thank you, Giuliano. Do you have any questions from audience? If not, we can give you 30 more seconds, if you can, for a question I have. I'm quite curious to understand what are the main benefits for farmers and policymakers using these technologies you mentioned.
Yes, of course. There can be different numbers of benefits. We can distinguish benefits according to farmers, to policymakers. For instance, farmers can, as I showed you before, thanks to this toolbox user interface, the users, for instance, can instantiate different simulations and make comparisons. This way also try to optimize the use of fertilizers, of pesticides, but also for policymakers, because there is another dimension, spatial dimension, which I did not show, for clarity and time constraints, which enable policymakers, for instance, at the regional level, to get some insights about what is going on, also to build this spatially explicit, somehow updates of what's going on. So the risk at the regional scale, so on using maps communicating clearly what is the spatial distribution of the risk according to specific pests or pathogens. And this, I think, is really interesting to change the way in which also policymakers, for instance, at the regional scale, try to manage this kind of information and risk.
Great, thank you for your answer. Thanks again to be here in presence also. So we can go ahead with the spoke number 3. We have also here in presence Roberta Paradiso, that is our associate professor in horticulture at the Department of Agricultural Science of the University of Naples, Federico II. So the spoke 3 is focused on enabling technologies and sustainable strategies for the smart management of agricultural systems and their environmental impacts. Today, Roberta is talking about a smart solution for precise agriculture and sustainable management of agricultural systems and innovative strategies for protection of natural resources. So please, podium is yours.
Thank you, Marco. Good morning, everybody, and thank you for coming. Just give me 1 second to close. Okay. Good morning again. And so this morning I will speak on behalf of Professor Luca Corelli Grappadelli, Scientific Coordinator of this book. This book is about enabling technologies and sustainable strategies for smart management of agricultural systems. And within the three strategic objectives of Agritech for a sustainable agriculture mentioned yesterday by Professor Ercolini, this book mainly focuses on the first two, promoting resilience to climate change and reducing wastage and the environmental impact of agriculture. And these objectives are pursued through three work packages with the specific goals. The first one is about smart solution for a sustainable agriculture and aims at developing a portfolio of smart techniques and technologies. The second one is about innovative strategies to protect the natural resources and to reduce pollution. And so it aims at developing, at promoting a sustainable use of water, soil, and ecosystems. And the third one is on demonstration to stakeholders to support the diffusion, the adoption of the proposed solutions. The spoke lineup consists of 15 partners, including 9 public universities and the National Research Council, and 4 private leader companies in the field of technologies for agriculture, and 1 private bank. And of course, each partners participate to the research activity with the specific expertise. Each work package is organized in different tasks assigned to the different partners. The first one, on smart solution, will include monitoring and modeling of crop, soil, and water based on innovative sensors, and also precise management systems and smart tools for automation of mechanization, irrigation, and fertilization, and digital solution for crop quality assessment. Some of these technologies will also integrate Internet of Things, artificial intelligence, and big data platforms. The second work package is about innovative strategies to protect the environment, and it will concern precise soil fertility management techniques and water use optimization, as well as the wastewater and nutrients recovery. And not only sophisticated technologies, but also nature-based solution for the environmental protection. The third and the last work packages will be about demonstration to stakeholders. This is crucial to promote to involve the stakeholders and to sustain the policymakers to develop the on-purpose rules. And it will imply the economic analysis of the proposed solutions, a multidisciplinary assessment of the new technologies, as well as the policy supporting innovation, and of course, last but not least, the involvement of stakeholders in a coordinated network. Let's see a few examples of our work. We mentioned that this spoke aimed at developing real-time, precise, proximal, and remote sensors. This is because to monitor several crop and environmental parameters. These parameters can include crop and soil water content or water status, as well as plant health and stress conditions related to biotic or abiotic agents. In this example, these sensors aim, this sensor aimed at driving irrigation in vineyards to improve the grape quality for enological purposes. And an in vivo probe is inserted directly in the stem to monitor in continuously the plant water status through the subflow change in terms of cationic concentration and composition. And this kind of device could help the farmers reduce the adoption of different and more complicated estimating method. Sensors can be also used for crop protection and in predicting the produce quality. For instance, multispectral or hyperspectral sensor can collect data related to the plant status, stress conditions, but also the presence of pathogen infection, but also the plant tissue composition. In this example, the analysis of RGB images acquired by an aerial vehicle was used in the early detection of a fungi infection and also in monitoring the bunch ripening. As mentioned before, also this system could alert farmers for in case of dangerous situations for the crop. In the view of a precise management and sustainable management of orchard and vineyard, the University of Bologna developed this robot, which is able to navigate even in these field areas where the GPS signals is not reliable because of the presence of different kinds of shields, including rain covers, anti-insect or anti-hail nets, but even in presence of photovoltaic panels. And thanks to a LiDAR sensor, and it is able to navigate and to maintain an accurate position respect to the plant's row, and to recognize the end of the row, and it can be equipped with a different kind of sensor to monitor the crop condition. With the same purpose, the University of Bari developed this robot specific for vineyards in presence of a plant with particular architecture. This is the case of plants grown at tendone layout and in presence of rain covers. It is an autonomous vehicle driven by electric engine, and it is equipped with sensors that can evaluate the condition of the canopy as well as the rate of fruiting. And it can be equipped, it is already equipped with a UVC lamp to irradiate locally with a high precision plant organs infected by pathogens. Moving from the open field to greenhouse, the University of Milan developed this autonomous and flexible platform able for both working open field and greenhouses. It is an autonomous vehicle equipped with a sensing system consisting in an RGB and 3D and hyperspectral cameras, and it also merges the working of this camera with some actuators. Indeed, a software analyzing these images can activate these actuators to distribute fertilizers, biostimulants, and protection treatments, also including novel molecules and UV radiation. Of course, the advantage is the improvement of yield and quality of the product, but also the safety, the food security of the produce, and the increase of the input use efficiency. And it is able to navigate even in relatively small greenhouses. In protected cultivation, our team in Naples works in a new strategy to improve both the efficiency of the use of the greenhouse volume and also of both natural and artificial lighting. In these vertical towers, different types of crops, including fruit vegetables or leafy vegetables, also tuberous plants or microgreens, are placed in the shelf from the top to the bottom according to decreasing light requirements. And each shelf is equipped with an LED artificial lighting system connected to a software that integrate the natural light reduced by the shading at different light intensity and different spectra, depending on the specific species requirement. And in the same research, some private partners also worked on the sunlight ray trace simulation to develop a model to predict the effect at the crop level of the movements of the sun during the day and along the season, as well as the shading effect of the greenhouse structure. In the view of the relevance of vertical farming and the need of monitoring the plant status during the cultivation, the University of Bologna developed this algorithm based on artificial intelligence to monitor the plant nutritional status based on the RGB image analysis. The algorithm was tested on basil, and the final result will be the development of an interface able to calculate in a very precise way the precise amount and type of fertilizer to be supplied to the plant in the different phenological phases. And this system will be also provided with an intelligent decision support system with a user-friendly interface. Agritech is not only sophisticated technologies, but also concern nature-based solution. I could not mention all the activities, of course, but before leaving, I just want to spend just a few words about the aim to protect the environment and the landscape. In this respect, some colleagues are working on a method to recycle wastewater in compliance with the European directives. And in some of the experiments, these wastewaters are used not only for commercial agriculture, commercial farms, but also in natural landscape. And this solution needs, of course, needs to study the effects of wastewater on the crops, on wild plants, and also specific drippers for low-quality water. This was a very brief summary of works done from many colleagues. I thank my colleagues for sharing their work, and of course, I thank you for staying.
So thank you, Roberta, for your effective presentation. So I was quite impressed by, I have a couple of questions for you, if you can stay, please. Thank you. I was quite impressed by your advanced level of maturity of the project that you are developing. So I'm quite curious in understanding which could be the contribute of your solution in terms of sustainability of our agricultural systems. So please, if you have 20 seconds, I appreciate so much. Thank you.
I will run. Thank you for the question. Let me say that it's not only my personal advance, because my duty was just to speak about the work of others, but my personal opinion is that our work, the work of research, is at a different level. I think most of the technologies we work on have already reached a technological readiness level that make them usable for the farms. But I think that we are at the last, at the most crucial step, which is the technological transfer. And so the next work will be involve stakeholders and policymakers and share with them the relevance and the importance for agricultural sustainability of these innovations.
Thank you. Thank you again. Thanks. So, we can go ahead with spoke number 4. Again, we have here in presence Alessandro Vannozzi, that is our associate professor of plant genetics at the Department of Agronomy, Food, Natural Resources, Animals and Environment of the University of Padua, right? So thank you to be here in presence, Alessandro. Spoke 4 is focused on multinational and multifunctional and resilient agriculture and forestry system for the mitigation of climate change's risk. Today, Alessandro will provide a general overview about the objective and strategy of this specific spoke, going through talking about next generation of technology and resilient traits of crop varieties and tree species, and next generation of genotypes and multi-omics approaches to predict complex resilient traits in crops. Is all? Yeah. Fine. Thank you, please. You have 10 minutes.
Non trovo. Ah, grazie, perfetto. Sì, che ci trova. Qui? Ok. Perfect. Good morning to everybody. Before starting, I would like to thank the organizer for the opportunity to be here and share with you part of our results within the Spoke 4 framework, and also to have the opportunity to visit Japan and visit Osaka and visit the Expo, which is amazing. So today I'm here as a representative of the Spoke 4, and in particular of the Task 411, which is one of the 11 tasks in which Spoke 4 is structured. And Spoke 4 deals with a multifactorial and resilient agricultural and forestry system for the mitigation of climate risks. The main goal, the main goal of our spoke is basically to create a sustainable and technological solution for smart agriculture based on two main principles: more with less and do not significant harm, which means that we have to try to produce more using less input, and we have to try to do it in a sustainable way, try to avoid to impact too much the environment. And to do that, we can work at different levels. The first level is working at the crop level, which means that we can use a genomic and phenomic platform to create database in order to produce improved varieties, resilient varieties, resistant varieties. And this is the first goal. But a second level of activity could be considered at the field level, which means using these crops, using these varieties or the forestry species or whatever, in a more sustainable way, which means improving the input use efficiency, basically. Our spoke is organized in 3 main work packages. The first work package is the next generation technology for resilient trait of crop varieties in 3 species. This package is more aimed at producing phenomic and genomic data to create resilient and resistant varieties. The second work package is more related to create agricultural and forestry systems and develop, integrate bio-based strategies to maximize the mitigation effects. And the third one is more aimed at creating models to predict the biomass production and the impact of climate changes on agriculture. So as you can see, the 3 work packages are organized in 11 tasks, 4 and 3 tasks for the last work packages. Today I will focus a bit more on the work package 1 and in particular on the task 411. I'm one of the co-supervisor and co-leader of this task, which is aimed at the next generation genotyping and the use of omic technologies for the molecular prediction of multiple resilient traits in crops. This task is composed of many different research units. We have 6 research units working on this particular activity. And as you can see, all the research units have different skills and different species in which they are involved in their research. We have researchers' activity in grapevine, peppers, eggplants, wheat, chicory, lettuce, olive, and also other varieties, like other species like tomato or legumes. Today, I would like to give you some examples, some of which has been used also as flagship solution that we produce within this task. Regarding grapevine, wheat, and chicory. So the first example that I'd like to share with you refers to grapevine, and in particular to the terroir. You probably have heard about terroir. I think we all drink wine, hopefully, and we all heard this word. But the meaning of this word is quite complex. Terroir is a sort of signature that the environment, but also the agricultural practices, the genotype that we use, exert on the wine and on the wine quality. And understanding how different variables, different environmental variables affect the terroir and the wine quality is particularly complicated. Why? Because there are so many different variables. And so to understand the impact of each variable, even in relation to the climate change, means trying to block all the other variables and to focus on a single one, which is quite complicated. Within the Agritech framework, we were able to set up an experiment which was quite complicated and was based on the use on better the transferring of different soils from different locations of Veneto region. And what we did was trying to grow 2 different grapevine varieties, which are particularly fancy for us because I'm from Veneto, and Corvina variety and Glera variety are particularly important in Veneto region for the production of Amarone wine and Prosecco wine. And we tried to grow these varieties in 3 different soils, trying to understand the effect of the soil on these varieties, and in particular on the berry quality. As you can see in this image, you can see different soils, different plants growing in different soils, and we estimated the impact of these soils on the phenology, which means how the plant developed, if there were differences based on this soil, but we also look at the biochemical effect of the soil, at the transcriptional effect of the soil, and in particular, we focus on the transcriptomics, which means how genes are moving because of the soil effects. Why it is important to identify genes that are moving because of the soil? Because if we know which are the genes that respond to the soil or to the microclimate, we can use this information to improve both the quality and the sustainability of agriculture, in particular of viticulture, and we can drive our selection of the best candidate varieties for a specific terroir. This is important for us. A second example regarding the PV varieties. You probably heard of PV varieties, or probably you drink or you drank PV varieties without knowing it. PV are resistant varieties which have been created breeding elite varieties like Pinot Noir, Chardonnay, whatever, with interspecific hybrid which are resistant to the major fungal pathogens, which are basically powdery mildew and downy mildew. Okay? And the problem is that we produce these varieties, but we don't really know which are the genes that we were able to introgress from the interspecific hybrid in the elite variety to make them resistant, because these genes are not known, basically. We only know the molecular markers that are close to these genes. And so we wonder, okay, are we able to understand at least what are the genes that can be associated to the resistance in these varieties? And again, we took advantage of transcriptomics, which is a branch of the genomics, in order to analyze the behavior of the PV varieties and the behavior of their noble parents to identify genes that can be used as target genes to improve other grapevine varieties, to transfer, for example, these genes in other grape variety, or maybe to edit these genes in other grape variety. So understanding the mechanism behind the activity and the resistance of PV varieties can help us to extend this trait to other varieties. This is another example, probably the most interesting to me, on grapevine, which is called Obiettivo 2100. What is Obiettivo 2100? Obiettivo 2100 is a population that we create. We created this population breeding together 7 different grapevine varieties, among which are Glera, which is Prosecco, Montepulciano, Sangiovese, etc., to create a big population constituted of approximately 2,100 plants. The fact is that all these different varieties are different between them because of, for example, the earliness or lateness of maturation, because of the extent of acidity at the body level, or the balance between sugars and acid. Okay? And these are all traits which are related to climate changes. Maybe not directly, but they're indirectly related with the climate change. For example, now we have basically, during the selection of grapevine, we selected grapevine because of the sugar content, but indirectly we selected grapevine for the earliness of maturation. And this is a problem now, because we have to harvest maybe half of August, or at the end of August, which are a lot of problems related to the safety, technical problems for the mechanical harvest, and so on. So we have to try to understand how to bypass all these problems. This population could be a solution, and this population is kind of unique, I would say, in the whole viticultural research system, because it's difficult to find population created by semi-diallelic crosses so big in grapevine research. This is the population we created. This was done in collaboration with the University of Udine. And when I would say that when I say we, of course, I'm referring to the whole spoke, not just to my group. And what we can do with this population is, by means of genotyping and phenotyping the plants, we can feed a sort of algorithm to create models. And these models can be used to predict the attitude, or better, the agronomic value of other progenies just based on the DNA sequence and not waiting for the field evaluation. This is particularly important for us. I will skip this one. Another example, we changed totally the species we are considering. This is chicory. Chicory, another important excellence for our country, for our region. We have so many different varieties of radicchio. Rosso di Treviso, Precoce, Tardivo, Radicchio di Verona, and so on. In this case, we were able to take advantage of the opportunities of Agritech to genotypize like 500 accessions of chicory, or better, within the Chicarium genus. And this is important for three main reasons. For the first one, enhance the use of land varieties and local crops. For sustainable breeding, if we know the markers, if we have genotypized all these accessions, we can plan breeding programs to create, for example, new hybrid varieties, and for the conservation of the genetic biodiversity. The final example I want to give you relate another species, which is wheat. In this case, we consider both durum wheat and common wheat. And again, we were able to, in collaboration with the CIS, which is the Italian Society for CIS, we were able to consider different accessions to phenotypize these accessions for the resistance to the main pest of wheat, which is Fusarium head blight, and to proceed with the identification with candidate markers, which were used to implement a panel, which is called Agritech, or better, Agrisec, sorry, and can be used to predict the attitude or the resilience affinity of wheat accessions in terms of production and resistance to this pathogen. These are just a few examples of the activities, and are examples just related to the task I supervised. There are many other examples. There are many different flagships that have been produced. I just focus on these ones because I'm more confident with these ones. But yeah, I would say that we are just starting our story, but the good things are still to come. And I thank you for your attention.
Thank you, Alessandro, to be on timeline. So I have just a quick question. So can you give us a shot, just a few words about the future of viniculture in Italy and also in global scenario?
In my opinion?
Yeah, your opinion.
No, I mean, future of viticulture, I spoke about our population, I spoke about conventional breeding of viticulture, but I really believe that the only way is editing. NGTs, TEA, we can call however we want, but editing is the easiest and probably the most safe way to guarantee a future in viticulture in the frame of climate changes. We all know probably what happened a couple of months ago at the University of Verona. There was the first experimental field where an edited Chardonnay variety has been planted, and unfortunately a couple of weeks later, all this experimental field was destroyed. We have to face also these kind of challenges, not only scientific challenges, but I really believe that that's the only way for us. And we are improving in many of the bottleneck to apply the editing to viticulture. So I do believe this is the way we have to move.
Thank you again.
Thank you,
Alessandro. Next two speeches comes from streaming, so are online, but I would ask to the previous speakers to stay on the floor, please, and then follow together these new two steps, two sessions. So next spoke is number 5, that is full focused on sustainable productivity and mitigation of environmental impact of livestock systems. So today we have online Professor Nicola Lacetera, that is currently the full professor of Department of Agriculture and Forest Sciences at University of Tuscia. Good morning, Nicola, and thank you again to join us so early. It's 5 o'clock in the morning. Today is, I remember to everybody that is Sunday, Easter, so happy Easter, Nicola. So today, Nicola is going to talk about solutions to improve sustainable productivity and promote resilience to climate changes, and well as resources, waste, environmental impact. So thank you, Nicola. You have 20 minutes. Thanks.
Thank you, Marco, and good morning, everyone, and also my best wishes for a peaceful and happy Easter. So I hope you can hear me. Yes, I guess you can. And so, very briefly, I want just to share with you the 2 strategic objectives the Spokes 5 is working on among the 5 strategic objectives of the Agritech National Center. So the first one is the enhancement of the sustainable productivity and the promotion of resiliency of the productive and livestock systems to climate changes. The second objective is the reduction of wastage and environmental impact of the livestock systems. As the other spokes, also Spokes 5 is organized in different work packages. In particular, there are 3 different work packages, number 1, number 2, and number 3. So just a few keywords which can help to identify the main activities carried out within each of the 3 work packages. The first one is relative to data, environmental, animal welfare, productive data, which are seen in terms of recording, management, and modeling by utilization of new technologies. Work package number 2 is relative to the improvement of resilience to climate change. And the third one is relative to the all the activities carried out to improve the sustainability, focusing on the environmental pillar within the 3 pillars of the sustainabilities, which are usually taken into consideration, even if we have some people, some groups working also on the economic sustainability of some solutions proposed within the Spoke 5. Well, different institutions are involved in the Spoke 5 activity. It is coordinated by the University of Tuscia, but also the University of Milan, Padua, Bologna, Pisa, Sassari, Naples, and Catania are contributing with different research groups. And we have also a private company, which is the Antares group. I will go straight to a short selection of the solutions different research groups are developing within the Spoke 5, and I selected a few solutions within each of the 3 work package. And the first solution I like to share with you comes from the University of Naples. The activities are being and have been and will continue to be carried out within buffalo farms. And the solution is called the thermal drop, and it is represented by a software for the automatic analysis and assessment of the correlation between temperature variation and production data relative to buffalo farms. So in the figure, you see an automatic milking robot and a weather station. So different data sources collected by different systems. Data are integrated, and the system is capable to identify animals more susceptible to climatic variables. And the integration with data coming from the milking robot permits the selection and identification of animals, which then can be and are being evaluated from a genetic point of view, permitting the genetic selection of more thermotolerant animals. So this is an example of data management and modeling finalized to adapt livestock systems, one of the livestock systems taken into consideration within Spoke 5 to climate change. Okay, let's go to a second solution. People from the University of Padua, Pisa, and from the Antares group are working and are developing. And yeah, what I can add is that all the solutions I'm sharing with you have to be considered around TRL between 6 and 7. This system is can be also considered still related to work package number 5.1, data recording, analysis, and modeling. And the system, and in a minute I'm going to show you a short video, allows detection, early detection of motor and abnormalities and nutritional variation in dairy cows, which represent two important aspects to be considered, in particular in the around the calving period, which is called the transition period between 30 days before till 30 days after calving. The system permits by image analysis a real-time analysis and video viewing, which is capable to allow early diagnosis of abnormalities for the locomotion of the animals or concerning the nutritional status. So I'm trying to share. This is the itinerary, it's the area covered by the animals moving from the milking area to the resting area, and there are cameras collecting and analyzing the image I'm trying to show you in a minute by this video. This is just, I hope you can see the video, and yeah, there are few animals passing from the milking area to the resting area, and which through the cameras are analyzed, and the system provides early warning on locomotion abnormalities and the body condition score of the animals. The system also permits to identify the single animals by the number on the ear tag, and also on the basis of the color of each of the animals analyzed. I think we are okay. So let's pass to a different solution, still relative to adaptation to climate change. Still relative to dairy cows, and the aim of this solution is to increase the sustainability of the cooling protocols, which are in most of the cases based on ventilation and water sprinkling. In particular, the system is aimed to contain energy consumption and water consumption. The system is based, it is an algorithm which permits to calculate a thermal index based on the value of temperature and humidity of the air, which becomes the temperature humidity index. So through continuous assessment of the THI, the system permits to establish the thermal balance, and on the basis of alerts which start when the heat load index indicates that animals have accumulated an excess of heat, the system permits an alert cooling of the animals. So animals are not cooled continuously, but only when the index indicates that they accumulated too much heat in their body. Okay, let's pass to another solution. This also comes from the work package dealing with resilience of the systems to climate change. And this is relative to extensive systems. In particular, the system based also, in this case, by image analysis, is based on an algorithm which permits recognition and classification of the typical theriofauna in the agroforestry systems, with a special focus on wild ungulates and the predators. The precision and the correctness of the system and of the algorithm is very high, in that it permits, it has a correctness of higher than 90% in the automatic identification of various species. I just want to underline that the importance to monitor the wild fauna is linked to health aspects, in that the wild fauna can veiculate virus or other microorganisms, which can be important in terms of health of the animals, but also in terms of human health. So it is an example of a solution helping to stress and to work within the framework of the One Health concept. The importance to monitor wild fauna is also linked to the fact that wild fauna can cause damages to animals or to feeds cultivated for animal feeding. The last solution I'm going to share with you is relative to the third work package. So the work package including research groups working on monitoring and mitigation of pollutants from farms. Also, this solution is relative to dairy cows or buffalo farms. It has been developed by a group of the University of Naples, and it is based on sensors installed in the bar or in the slurry storage tanks. And the sensor permits registration of the concentration of some pollutants in the air, in particular ammonia and CO2. And the systems is based on an IoT platform which permits in continuous visualization of data, which can help to farmer also to evaluate the effectiveness of mitigation targeted action which farmers can put in place to reduce the environmental impact of their activities. So I thank you very much for your attention. I'll be glad to answer to questions which can come from the audience.
Thank you, Nicola. Thank you. Any questions from the audience? Okay, so thank you, and have a nice day, Nicola. Thanks again.
You too. Bye.
So we go ahead with the next speech that comes from spoke number 6, that is focused on management models to promote sustainability and resilience of agricultural systems. So today we have online Luisella Celi. Please give me a feedback about the presence online. Luisella is full professor at Department of Agricultural Sciences of University of Turin. Here you are, Luisella. Thank you to be here, and good morning again. Happy Easter also to you. So today Luisella is going to talk about the exploration, cutting-edge strategies to enhance sustainability and resilience of agricultural production system. So thank you to be here, and go ahead. Thank you. You have 20 minutes.
Good morning, everyone, and welcome to this presentation. I'm very pleased to show the results of this SPOC, which focuses on finding management models to promote sustainability and resilience of agricultural production systems. Our SPOC include different partners from University of Turin, Università Cattolica del Sacro Cuore di Piacenza, Politecnico di Torino, Università di Genova, Università Politecnica delle Marche, Università di Foggia, together with 3 private companies, Relatec, De Matteis, and Graded. Our SPOC is organized in 3 main work packages for the development of these models. The first one is aimed to promote sustainable and resilient agricultural management models supported by data-driven decision support systems. This includes the development of practical indicators that can be applied in agriculture and are validated using case studies. The second one is related to determine the environmental, productive, and economic assessment of circular models that utilize bio-waste, and this work package includes the validation of circular systems and the evaluation of bioconverted materials that can be used as biofertilizers, biopesticides, compost, and biostimulants. And then the third work package is strictly connected with industries that are functional to agriculture to arrive to develop an economic and socio-cultural model that is rooted in the integration of enable technologies in agriculture. So this approach derives from an evolutionary process that began with conventional high-input agriculture. Over the years, the production, as production priorities shifted from a strong market-oriented focus to increasing concern for environmental sustainability. So the agriculture paradigm has transformed, and today this evolution is reflected in the emergence of a new integrated model that combines principles of preservation, efficiency, and sustainability. So we arrive to define it in a new word that is, and in a new model that is regenerative agriculture. But what is regenerative agriculture? So we give a new description, such as an updated and holistic expression of agriculture that incorporates the approach of organic farming, carbon farming, conservative agriculture, but with the adoption of the best technological innovations that we have in the market, and we apply for nutrition, for plant protection, and for the genetic improvement in plants or in animals. And this approach is closely aligned with the society expectation and market demands, providing also environmental sustainability and agricultural productivity. So we have different points that are combined in these models, leading to a flexible model that should be adapted to the different needs of agriculture and of farms. And in this presentation, I will give some example of what this regenerative agricultural model can be in terms of digitalization for carbon farming, water and nutrient management, and circularity. Let's start from digitalization. Digitalization is really important in agriculture because agriculture must undergo a digital transformation involving continuous monitoring, data collection, and data processing. This is essential to develop indicators and models that feed into decision support system, helping farmers make more informed and timely decisions. To enable this, we have developed a digital platform with a standardized procedure for data collection, transfer, and storage, collecting all data that control an agricultural system. And this platform addresses the challenges posed by connectivity gaps across different regions. So as you can see in the map, we have many stations across Italy to have a very good pedoclimatic transit and collect data that can give information based on the difference between the various agricultural farms that we have, for example, in lowland, in hilly, and in mountains. So considering the different size of the farms. And data collected through this system have also enabled the development of predictive models. Here we give an example developed by the University of Piacenza that allow to associate the main alternaria species that can be present in tomato crops This model captures weather data, defines the biological processes of the pathogen, and considers plant phenology to simulate and anticipate the dynamics of disease. Another part that we are developing in our work package is carbon farming. Carbon farming drives agricultural practices toward more sustainable and regenerative models based on data evidence. So this means that we have collected many technological innovations to indicate how farmers can increase the carbon stock in their agrosystems. But we need to have advanced monitoring system and intelligent management. So this means decision support systems to track the stock of organic carbon and show the different level and show if the system they adopted is working or not. So this means that we need this MRV protocol for soil carbon. What is MRV? This is a monitoring, reporting, and verification system that configure and enroll farmers enroll farmers, and then once the farmers are adopting these technologies, we have a system that monitor and measure the variation of carbon. But what is very important is the report and the verification, because finally all collected data are consolidated into a comprehensive report which is verified by an independent third-party entity. This step ensures transparency and credibility, both of which are essential for the legitimization of the generated carbon credits. So in this way, we can arrive to define if a farm really produce carbon credits. And here you can see how the system works and how we can define the stock baseline in terms of carbon using remote and proximal sensing. We can determine the optimal soil sampling strategy to minimize cost while ensuring accuracy. And we identify all phenological steps, and we start from crop sowing, and we arrive to collect harvesting dates for input into soil and biomass models. And then at the end, we can quantify the amount of crop biomass and carbon returned to the soil. Another aspect we wanted to show is water management, very important in this particular period. So also in this case, we developed new models, agrohydrological models that can simulate irrigation water demand during the growing season as a function of different factors such as soil properties, climate, crop needs and characteristics, and the current irrigation systems that are used. And we can estimate using this model both the water demand in real time and based on future scenarios. For example, in this map, we have the water saving that we can obtain using only micro irrigation compared to the current irrigation system. And as you can see, we can have saving water percentage ranging from 0 to more than 60%. But of course, these new technologies that can be used to water for water saving should be tested to meet the crop productivity. So we tested different techniques. What we observed that with the fertigation or micro irrigation, we have a higher productivity compared to flood irrigation. But we have also to consider that we can have a secondary effect. For example, fertigation must be carefully evaluated because we observed that this type of irrigation can determine the contamination of mycotoxins. And then we have another pillar of our SPOC that is circularity, because we can have models that can create a very virtuous system. For example, we worked on connecting not only livestock farms, but also agro industries and orchards and vineyards residues to plants that can provide the transformation of bio-waste. For example, here we have the connection of different systems that confer bio-waste to an anaerobic digestion plant that is able to produce biogas, and this biogas has a very big upgrade because separate biomethane from CO2, and this CO2 is used for producing the gasification of water. So it's very curious that we can use the carbon coming from animal manure for producing sparkling water. And then we can also upcycle the byproduct of an anaerobic digestion plant, the digestate, that is in part returned to farms, but in part can be treated, transformed in compost, but also in biofertilizer, struvite, that is a fertilizer, nitrogen phosphorus fertilizer, biostimulants, and in this case, water is also separated and returned to water bodies. And another upcycling of this system has been the production of nutraceutical materials and food additives that can have a very high added value. And another example in terms of circularity is the use of black soldier fly larvae.
Rosella, excuse me if I interrupt you. You have 1 minute, please.
I finished. Of a black soldier fly that can be used for the animal feed. So is this society ready? The question, the most frequent question is, I don't know, both for consumers and farmers. So what we are doing is to create a living lab with many actors, consumers, farmers, policymakers, to build the regenerative agriculture. So thank you very much for your attention, and I'm open to questions if you have. Thank you very much.
Thank you, Luisella. to join us this morning. Any question from the audience? So thank you again, Luisella, and have a nice Easter.
Thank you. Bye-bye.
Bye. We can proceed with Spoke 7 that is working on integrated models for the development of marginal areas to promote multifunctional production systems, enhancing ecological and socioeconomics sustainability. So today we have here in presence the speakers. I get the opportunity to switch the floor, thank you to Giuliano, to Roberta and Alessandro to be here today, and I say welcome to the next two speakers. Cinzia is with us. Thank you. Good morning. Good morning. And then we have also Antonio Paparella. Please take the floor. Good morning, Antonio. So, Spoke number 7. Today, Cinzia is talking about evaluation of Italian marginal areas to define specific needs in terms of land management and soil conservation. So, Cinzia is associate professor at University of Bari Aldo Moro. Thank you, Luisella. Please.
Don't worry. Okay, thanks to everybody, thanks to the organizer, and of course thanks to the Ministry of University and Research to support these important and fundamental projects. So, let's start with Spoke 7. The Spoke leader is the University of Bari, and you can see that in this Spoke there are 8 universities and 4 public and private companies spread in all the Italian territory. So when we started to write the project about the marginal areas with Professor Maria De Angelis, we understand that marginal areas are not so marginal because the 70% of Italian territory is covered by marginal areas. So it's very complicated to define what is really a marginal areas because we have different kind of situation. So marginality can be done by natural situation. So we can have sandy soils, rocky, shallow, sloping, and so a limited reservoir of nutrients. We can have also adverse climate conditions. But marginal areas can be defined also for the human activity because, for example, soil salinity or alkalinity, erosion, loss of organic carbons could be realized by human. And also are important to consider the socio-economic constraints, so limited access to markets, scarce cooperation among farmers, especially in the south of Italy, and also bad infrastructure. So, in this way, we are thinking about the development of 4 work packages that you can see in this slide, and every work package has a specific goals. In my presentation, I will show you some of the solutions that can be defined as development of these marginal areas, but you have to consider that with marginal areas we introduce the mountain, for example, areas and all the coastal areas. So the solution is very different because are very different the territories in which they refer. First of all, this solution that is named soil scoring by DNA analysis marginal areas is a protocol development to define indicators to assess functional agrobiodiversity and quantify ecosystem service. So in which way this protocol will function? So extracting DNA from marginal soils and quantify specific microbial genes of the microbiota, the life part of the soil. In this way, by the quantification of these genes by real-time PCR, we can have an idea, a precise idea on the health of the soil. So for example, in this slide, you can see that some soils needs and fixing, inoculating. Other are high fertility. Other are still good for wheat. So with this mapping and with analyzing by real-time PCR, we can develop the best solution for the soil of marginal areas. Another important thing is the phytoremediation. Phytoremediation is a technique in which some plants could detoxify some soils contaminated with toxic elements. So in this way, we develop phytoremediation protocols, new technologies for rapid monitoring of the remediation process. In this way, we can recover soils that are underutilized because are fully of toxic elements, and combining different techniques like hyperspectral camera and micro X-ray, we can define the best solution to detoxify soils. Here you can see an example of the soil polluted before and after restored upon the incoming generation of plant biomass. Another approach that we studied was the use of X-ray microanalysis of forestry cores, because of course also forestry are considered marginal areas. So we have two opportunities to use this technique. One is non-invasive approach, so the use of micro X-ray fluorescence, a high resolution, rapid and no destructive techniques to study the presence of potential toxic elements in forestry. So with this technique, studying the guttation fluids of the forestry trees, we are able to discover which contaminants are present in the xylem of the plants without a disruptive approach. Another important result of the project was the construction of living lab. Usually, living labs are focal point in which all the people that are interested in a topic can have a discussion. In this way, we constructed really a living lab internal to the campus university, because this living lab representative 13 thematic areas, all the environments that are included in marginal areas. In this way, students, citizens, and people that doesn't understand the importance of this area can really see what is a marginal area, and there is also a place in which we can have discussion and meetings open air. Another problem is the animal resources. As you know, farmers, we have a problem with farmers to have cooperation and to utilize in the best way the animal resource. In this case, we studied the use of sheep and the production of wool, because there is a strong request of this material, but there is no an organization to provide this. So in this project, we studied a protocol to create innovative methodologies in the data collection, and also to help the farmers of the sheep productions to have the best solution to guide the genetic selection of this animal, because a big problem that we saw is the inbreeding coefficients that is too high, because usually animals are crossed together, and so there is no organizing in this way. Another topic is the usage of treated municipal wastewater to irrigate olive and almond. Olive and almond are two typical crops of South Italy, and they are rustic plants, well adapted also to marginal areas. So we focalized our attention on the experimental plot using this kind of recircular water coming from municipal wastewater, and we investigated the physiological response by omics technique, so in this case RNA transcriptomic technique, so to discover which kind of genes are activated in olive and almond, and also if the production of these plants could be suitable with this kind of use of wastewater. Another focal point for the development of marginal areas is the biodiversity. Biodiversity is a challenge in this kind of area because we have a lot of abandoned territories and a lot of crops, varieties, at risk of genetic erosions. So we pointed out a protocol combining different aspects of economic, genetics, and agronomic to discover the potential of some crops like grape, almond, olive, and other fruit crops, and in the same time to discovery and recovery the landscape of marginal areas. Because we don't know, but in this area could be very high important cultivars that are key for the preservation of genes that could be useful to the breeding programs. For example, in Italy, in Apulia, we have the problem of Xylella fastidiosa, and we are finding a solution, also investigating these ancient genetics variety. I want to go out, but because there is the other slide, but it doesn't work. I was just at the end. And it's stopped it on this. I don't know why.
Someone from the staff can help on this presentation. We have some problem on switching this slide. So.
Okay, so another focal point is the valorization of plant resources. For example, in this solution, colleagues developed a system to use a halophyte plant that is called Salicornia, that is a plant that could be used, for example, to add in different preparation instead of salt. And it's very important because it's able to grow in soils with a lot of salinity. Another important if it works, but I remember, is the use of waste products like almond, carrots, and grape, another solution to extract nutraceutical compounds, because in this area you can also obtain important elements from waste, agricultural waste, and extract these components and add to foods. That's all.
Thank you, Cinzia. Also, without the last,
I was remembering
slide, you got the end, so thank you. And that's fine. Do you have any questions for Cinzia? Okay, thank you, Cinzia. Thank you again. We are approaching the end of this long session, so this morning. So I would ask to Antonio Paparella to take the podium, please. So Antonio Paparella is postdoctoral researcher that is involved in the Spoke 8 that works on new models of circular economy in agriculture through waste valorization and recycling. Today, Antonio will talk about how to transform agricultural waste into valuable resource while promoting sustainability across the support chain. Please.
Can I have the presentation from the laptop? Is it possible?
Sì, sta sul PC qua. Sì, sì, è già in modalità presentazione.
Okay, yeah. Again, my name is Antonio Paparella. I'm a research fellow at the Naples University. First of all, I want to express my gratitude. I'm such honored to be here and present and represent the Spoke 8 and to be able to show some of the results that we have achieved during the project. It's still not working here. Okay, but I will use the presentation here. Okay, perfect. So probably some of you are already familiar with this infographic. Those are the 5 strategic objectives of the whole Agritech project. And the Spoke 8 contributes to 3 of them: to reduce wastage and environmental impact, to develop a circular economy strategy, and to promote sustainable development of marginal areas. As the name of the Spoke suggests, circular economy is the core framework guiding the research in the Spoke 8. Circular economy is an expanding framework with a growing number of papers published each year, and you can see from the graph that a consistent part is linked to agriculture, indicating and highlighting the strong relationship between agriculture and circular economy. The Spoke 8, as also the other spokes, is structured into work packages. There are 4 work packages. The work package 1 focuses on developing new approaches and technologies to extract high-value biomolecules, polymers, and biomaterials from organic waste. These materials can have both biological properties and technological application. Then there is work package 2 that promotes the sustainable agro-energy production by valorizing wastes through biological or thermochemical processes, while still avoiding any conflict with food or feed production. Work package 3 develop and test and validate innovative technologies to produce fertilizers in order to reduce agrochemical use and enhance soil fertility, while still mitigating climate change. And the 4th and last work package aims to develop an holistic approach that ensures the sustainability, circularity, and an effective integration with the agricultural activities. In all these work packages, there are dozens of university involved, many research agencies, and also many companies. And so there are around 1,000 people involved in just this spoke. I will now pass to present an example of the results that I've been producing during the work. And as I said, the work package 4 is involved in the study, the integration of the technologies back to agriculture. If we see the infographic, during the work of the other work packages, wastes are upcycled, are reused, are used to produce energy, for example. But then after the process of energy production, there still is some byproducts, biomasses, that needs to go back into the agricultural sector in order to close the cycle and to pursue a real circular economy model. And this last step is often the most overlooked because it can be achieved just if farmers agree with that, and they play so a central role in closing the circle. The name of the work that I'm presenting is the study about the drivers of adoption of circular innovation with insights from field experiments on olive growers. And so the case study is the olive oil supply chain in the Apulia region. Apulia region is the lead producer region of olive oil in Italy, and Italy is the second world most important producer of olive oil after Spain. And with this huge production, around more than 1,500 tons of olive produced yearly, there is also a huge production of the resulting biomass, the olive pomace. More than 60,000 tons of olive pomace are produced yearly. The olive pomace is the resulting biomass after the olive pressing. The technology that has been developed during the Agritech is the usage of this biomass as the fuel for anaerobic digestion. And so there is the production of biogas, energy, heat, and also the remaining digestate after the olive pomace undergoes the anaerobic digestions. The objective of this study was to measure the willingness of olive farmers to adopt the use of digestate derived from olive pomace as a soil conditioner. That's because the digestates, in respect to the fresh olive pomace, has much better characteristics in terms of soil erosion prevention and to enhance soil quality. And the second objective was to identify possible drivers or also barriers to this adoption. We have conducted a questionnaire through olive producer of the area where the plant is already operating, so in Apulia region. And we collect several information, among which also their willingness and intention to adopt this innovation. The results are quite promising in the sense that around 70% of the samples of the farmers that were interviewed indicated an intention to adopt the technology. While on the other hand, just only 15% of the sample firmly opposed the adoption. So, all in all, there was a satisfactory level of adoption intervention. But this was just the first step of the analysis. The second step was to take this intentional adoption and use this as a dependent variable of an econometric models. The econometric models that are being used is the ordered probit. it's the model that you use when your dependent variable is an ordinal categorical variable. And this allows us to test the influence of different drivers of adoption that you can see on the right side of the screen. So technology push, managers' environmental awareness and manager risk aversion, market pull factors, regulatory push and pull, and trust in institutions. Results of this study were that there were some drivers and barriers, more specifically technology push, managers' environmental awareness, and regulatory push and pull. More specifically, we highlight that there is a negative effect of external advisory service, and maybe because farmers that delegate their decision, their agronomic decision to advisory services, thinks that the decision to adopt or not to adopt the use of digestate is one of the decisions they want to be delegated to other people. And we also highlighted a positive effect of subsidies on the adoption, and that leaves rooms for a policy intervention. And also we highlight the positive effect of farmers' environmental risk awareness. So still the psychology, the attitudes of farmers play a key role in accepting the innovation. And with that, I end my presentation here. Thank you for attention.
So thank you, Antonio, to stay on timeline. So I got 3 points from your presentation. You talked about new products, if I got well, agro energy and management of nutrients, right? Mm-hmm. So yes. I'm quite curious in understanding which of those is the most closely connected to the farmers.
Okay, I think that the third that you mentioned, so the production of the new fertilizer or new way to approach the fertilization is for sure the closest to the agricultural sector. That because every farmer, from the smallholder farmer to the big farm, have to have a fertilizer strategy, and so this point regards every farmers.
Great, thank you. Thank you again. Any other questions from audience? If not, we can proceed with the last, thank you again. We go ahead with the last speech that comes from the streaming. Is it streaming? Give me a feedback about the presence of Angelo Riccaboni. Yes, it is. Good morning, Angelo. Good morning and good Eastern. Good Eastern to you.
Good morning to you.
Good morning. So Angelo Riccaboni is a full professor of business administration at the Richard Godwin School of Economics and Management of the University of Siena. So he is the spoke leader for the Spoke 9 that take care on new technologies and methodologies of traceability, quality, safety, measurements, and certification to enhance the value of protect the typical traits in agro-food chains. So today, Angelo is going to talk about how tracing and certifying sustainability, origin, authenticity, and safety of agri-food products. Please, stage is yours, Angelo.
Good morning, everybody. I hope that you can hear me well. So I apologize for not being with you in Osaka today. And I will talk about the results of the spoke. It is a spoke which is mainly concerned on basically how quality, sustainability, authenticity, connection with the territory can be really a lever for competitiveness and profitability of farmers. And this is something very important, especially for Italian agricultural productions, where we have several value chains, several products, and the secret of the products is their connections connection with the territory, is their quality, is their authenticity. So what is very important is how to support our farmers, our food companies in their activities. Also because our companies usually are very small and they need support in terms of tools, in terms of solutions, which are able to valorize their activities. So today I will talk basically about 4 points. The first one is traceability, what we are doing on traceability. Secondly, what we are doing on measurement of farmers and food companies' sustainability. Third, the development of an integrated and truly reliable information database on food system for citizens, institutions, and policymakers. And also, everything is connected, everything is integrated on a new tool, a new major solution, which is called Metrica. So let's start with the first issue, traceability. Traceability means many things. What I'm talking about is the traceability of the geographical origin, which is one of the major topics because, as I said before, our products are of big value if we are able to be sure, or we are able to ensure to the consumer that those products come from the area they are declaring. So it's very important to be able to be sure that if I see that some cheese comes from an area, or some vegetables or some wine or some olive oil come from a certain area, they certainly come from that area. So for instance, I refer in particular to the several matrices, the several products where we are focused on, which are olive oil, wine, cereals, fruits and vegetables, and dairy. So what they are doing, what our researchers, what we are doing, is to find ways through experiments, through scientific approach, so through not documents, but chemical, genetic analysis to be sure that this product comes from that area. In particular, if you consider what we are doing on extra virgin olive oil and wine, we are analyzing soil, leaves, olive fruit, and extra virgin olive oil, and we do a lot of chemical and genetic chemiometric analysis, and we take all the results, and with the multivariate analysis, which means through artificial intelligence, we arrive to define where the olive oil comes from. And also the other similar approaches follow for wine. So again, soil, leaves, grapes, and wine, and then there are some analyses which are allowed from the laboratories that we have developed thanks to the Agritech Spoke 9 resources, and taking all this data, analyzing them with the artificial intelligence, then we have the results. Here you have some examples. I don't want to go into the details, but it is very clear that we are able to even allocate a certain product to a very small area. Even within the province of Siena, we can connect production to single small areas. So we are very satisfied by these results. The same with wine. So several analyses, we put them together, and we have a very interesting results. So we are doing, of course, also some blind tests. For instance, we have a wine which comes from a very typical Toscan grape, which is Sangiovese, but we analyze Sangiovese from Tuscany, Sangiovese not from Tuscany, not Sangiovese from Tuscany, not Sangiovese not from Tuscany. These are 4 categories, and we were able to find all of them, where they come from. While we deal with the traceability, we also consider the traceability based upon documents. And what we are doing in this field, we know there are some peculiarities of traceability for the farm. It's not an easy task. What we are doing, we are supporting the farmers to find ways to check which is their best possible solution. So we know that in the market there are several possibilities, several solutions, several tools which are at disposal of the farmers. But what we want to do is to better understand and to give the farmers the possibility to choose the solution that it is more suitable for the needs of single farmers. So we have we have developed an assessment tool able to do that. The second pillar, the second major area of activities is the measurement of farmers and food companies' sustainability. This is very important because more and more farmers are requested by consumers, by institutions, by regulation, by large distribution organizations, by investors, and by banks. Banks play a major role in that. So all these actors are asking to have more rigorous information about the sustainability of the company, the sustainability of the activities of the farmers. So what we did is a platform, a digital platform, through which a farmer, a food company can measure its sustainability, and also we support them in reporting about their sustainability with different also levels of completeness. So we can do a very simple report for those companies or those farmers which are at the beginning of the journey towards sustainability, or where their internal culture is not that in line with management systems, with the issues of good management, because they are very, very small or very simple. So we have also different reports that we can support to prepare. So at the end of the story, with this tool, farmers and agri-food companies can produce their own sustainability report. But also we have prepared, we give the farmers the possibility to assess their sustainability through a rating system that we have created. Last but not least, which is very important, is to introduce farmers to the calculation of their GHG emissions. So this is extremely important because, as you well know, one of the future evolutions in farming will be to take advantage of the market of credit, of carbon credits. So in order to enter the market of carbon credits, you need to measure your GHG emissions, and farmers, many farmers, don't know how to do it. They don't know which is the right path to do it. So we support them in this growth. The third major pillar and this flagship solution that we are presenting to you is a database, which is a database able to collect integrated and reliable information of food systems. I say integrated and reliable because, as everybody knows, on the web we have so many data about the food systems, but they often are not reliable. There are a lot of fake news. There are there's a lot of rubbish in the internet about food systems. But also this information must be integrated one with another. So AgriHub is the digital solution able to collect many data which are coming from reliable sources and give the user the possibility to build graphs, reports, to build what are, even with the artificial intelligence, short reports, short descriptions of the topic. So this is a very important tool. We saw that it is much appreciated by our stakeholders. It is regularly updated with an open access philosophy, user-friendly, ready to use. And as I said, we can play, we can work with this data, with this analysis, tables, and more than 70,000 reports are allowed. So also we have a chatbot which can be interviewed, and the chatbot will answer to the requests. Sources are only reliable sources. Sources are official sources. For the first time, they are put together, and as I said, they are integrated, so we are sure that the answer that which comes, for instance, what can we say about cereals production in Tuscany? And you have in a few seconds all the data about cereals production. If you want to know imports of a certain topic, you have the answer. If you want to know which is the production of olive oil, which is the production of any product in any area of the country, you can have the answer. So we see as a very important tool in order to support decision makers, institutions. For instance, when they want to make an investment in the area, they can have with this tool a clear baseline in order to better understand which is the impact of their activities. Fourth is the development of a data space for Italian agri-food. So what we are doing, this is another major achievement that we have prepared, is to have an integrated solution, which is, first of all, hosting all the tools that I presented to you so far. These tools are put together, they are presented, they are available through this Metrica data space. But more on top of it, I would say, on top of it, also, this data space is very useful in order to connect in a better way different actors in the food system. And this is very important because very often if we want to trace productions, if we want to follow which are the processes of a company, we do not have this possibility to connect different actors. So this is able to contribute, this Metrica, this solution contributes also to the development of some decision support systems, VSS, for instance, with reference to the grape supply chain, putting together all the actors and different tools, we are able to estimate the yield of the production, but also other important identification targets can be achieved thanks to this integration. So, in short, going to conclude, again, just to recapitulate what was presented to you today, we are very pleased that we are working hard on a topic that is very relevant for our farmers, for our food companies, and the topic is how to measure, how to ensure to the consumer, which is the quality, which is the sustainability, which is the authenticity. We want to ensure which is the connection with the territory. The connection with the territory is a key value for our productions because our products are famous and sold all over the world, but the consumers in Japan, of course, you would like to know if really that wine, if really that olive oil, if really that cheese is, or that oranges really come from the area they are declaring. So this is very important in the protection of the consumer, in the protection of the commercial value of the products. So the traceability is a key issue for us, and we are very, very keen on it. Secondly, we want to support farmers and food companies in their relationship with the market, with the consumers, with the banks. The banks are asking more and more information about sustainability because they are pressed by international institutions. So if we don't help farmers cannot produce this information. But also we want to give stakeholders in general more integrated, more reliable information of food systems, and they can do it through our Agri-Hub solution. Fourth, we want to create a data space for our companies in order to be sure that the use of information, the production of information is used for their purposes and to support their competitiveness. So in short, our food companies, our agri-food companies, our farmers need support in order to deal with the challenges in front of them, especially in the journey towards sustainability. And Agritech and Spoke 9 are working hard in order to do that. I take this opportunity to thank the chair of Agritech, Professor Lorito, the scientific director, Danilo Ercolini, and the general director, Marco Pacini, for the support they are giving for achieving our results. Thanks a lot, and then back to you, Marco.
Thanks, Angelo. Great presentation, very effective. Thanks again for waking up so early. So, any question from the audience? This is the last speech. So this was the last speech. So thank you again, Angelo. So wrapping up what we say today, we are approaching the end of this long morning. So just a short overview and wrap up of what we said today. So we started with the project overview and presented what the hub activities are in charge of. So then we passed through the spokes and activities carried out by the spokes. The spokes number 1, Novels tool for biodiversity of genetics resources. Number 2, focused on spatially explicit decision support for crop health prediction. Then the spoke number 3 is for that works on a smart solution for precision and sustainable management of agricultural systems and innovative strategy for protection of natural resources. Spoke 4 that presented the next generation of technology for resilient traits of crop varieties and tree species, and next generation of genotype and multi-omics approach to predict complex resilience traits in crops. Spoke number 5 talked about a solution to improve sustainable promoting and promote resilience to climate changes. Spoke number 6 that presented exploration of cutting-edge strategies. Spoke number 7, evaluation of Italian marginal areas to define needs in land management and soil conservation. And then the spoke number 8 that talked about how to transform agricultural waste into valuable resources while promoting sustainable across the supply chain. And finally, the spoke number 9 that we just followed that talked about tracing and certifying sustainability, origin, and safety of agri-food products. This just to say that we flow over the project in this long trip. This is just a portion of what we are doing every day here. So I hope that we transfer all the multidisciplinary that we are in this project that is really complex. The complexity of this project was probably never approached before in the past. So we did a great job, a huge job. We have in 2.5 years, in less than 2.5 years, we have in front of us around 10 months that are really challenging. We need to be really strong in reaching all the targets that we have on this project. We are full confidence on this achievement. So what we can say, with all these technologies, we receive from the project a huge heritage, a great, let's say, treasure that we have to use in full in the future, because this is the base to achieve a competitive advantage in these industries. So it's a really strategic project. So next 10 months are challenging, but provide the future for the next sustainability, technical sustainability for the next 10 years. So thanks again to everybody, for all the speakers to be here today, and happy Easter again to everybody.